So this next group of mollusks is the bivalves, and as you know from your lab work, bivalvia means that it has two shells, and those two shells are highly protective. They also create some challenges in terms of mobility. But this is the bivalves. They also are called the pelecypods. We know that the poda means foot, but the pelecypod refers to the shape of the foot, and that peleca in the Latin means hatchet. So these are the hatchet foot organisms, also known as the bivalves. So you know the term bivalve. We also know that these have bilateral symmetry, right? We've seen that in the lab. We also know that they feed as filter feeders through in-current and ex-current siphons. We'll talk a little bit later about how that happens a little more carefully. We also look at the fact that these tend to have really large gills. You can see that idea right here. These are the gills right here, and for the size of the organism, they're quite big. So just as a visual, two valves, bilateral symmetry. This particular group of bivalves here, the clams, they have this large foot, comes out of the base of the shell, the opposite end of the shell from where the siphon occurs. So here's our siphon right here. Here's the foot down here. The foot is for digging. The siphon is for extending up and filter feeding. You can see in all of these, here's our foot there. This is a particularly interesting foot, this spinal cockle with a red foot. And here is a surf clam extending its foot down into the sand. So this is how it burrows down into the sand. So the next question is, how does it actually use that foot? Well, it's a muscle, and muscles really only know how to contract in terms of the muscle fibers pulling on one another. But this particular organism has the ability to fill this structure, the foot structure, with blood. It fills it with blood, and that extends the foot out. It creates a little hatchet shape right here at the base of the foot. That expands into the sand, and then the muscles contract, and they pull the clam downward towards the extended foot. So they reach the foot down, extend, and pull the whole clam body down into the sand towards it. So here we have a slide that addresses the feeding process of the clam. We know that they're burrowed down into the mud. Filter feeders like the mussel will do the same kind of thing. Even scallops, although they're either attached to hard substrate or perhaps not sitting down in the mud but on the surface of the substrate. So all of these organisms have an in-current siphon through which they take water in, and they have an ex-current siphon out of which comes water that's gone across their body inside the shell. So they sit near the surface, or they sit exposed to the water. They filter through the in-current siphon, and notice the little frilly edges right here. Those frilly edges are for making sure that bigger materials don't get coming into the siphon. The material gets drawn into the clam body, food gets extracted, you'll see how in just a second, and then the water comes out the ex-current siphon. So they have the ability to extend this siphon very, very long ways, some of them up from where they're buried in the sand all the way up. Other organisms like a mussel, they have much less extensible siphons. From the inside view, here is the inhalant or in-current siphon right here. It draws the water inside, containing bacterial particles, plankton, other materials. That water flows really across the gills is what we're really interested in, and those gills are sticky and pleated like an accordion with lots and lots of surface area. You can see the pleats right all the way across here. The food particles get stuck on the gills, and then those gills trap the food particles with some mucousy layer, and then these palps right here, these are labial palps right here, those palps will reach out across the gills and rake the gills, moving the materials towards the mouth region, and so they'll rake it in, feed themselves, and then the water exits out in this direction, coming out of the organism, having had the food. Here's a better picture of the palps right here. The mouth area is just behind that, and the palps reach across, they rake the gills, put the food into the mouth. So here too, we need to think about how form matches lifestyle. So we have this idea, the concept of form. How does it match lifestyle? Lifestyle, these are sedentary, oftentimes burrowing creatures. They don't move fast. They don't pursue their prey. They sit there, water comes to them, they feed that way. So protection is the biggest thing for them, and the ability to either protect from predators with a really, really strong, hard shell, as in this one here, or the ability to dig quickly into the ground to escape a potential predator is what we see in both the razor clam here and the gooey duck here. Both of these are highly able to dig and therefore escape predation. We also see none of them have a head region, right? None of them have extensive separate sensory apparatus that sticks outside the body, so they are really built for slow, sedentary lifestyle. They have to protect themselves and get food in other ways aside from pursuit and escape. Given that they're sedentary and slow moving, what we see here is, as we mentioned in the beginning video, an open circulatory system, which is indicative of a sedentary lifestyle. So here we see gaps between the blood vessels. Here the blood goes out into sinuses to deliver the oxygen, pick up the CO2. It's not always inside the capillaries. So we go back to the idea of form matching function. Given that they're mostly sedentary, they don't have a head region, most of what they need to sense, they sense from the edge of the mantle here. So on any of these organisms, they have concentrations of sense organs on the edge of the mantle. This one shows us that the scallop has eyes as well as tentacles on the edges of the mantle. And if we look down here at the diagram, we see this idea of a pedal ganglion. That means it has a concentration of neurons in its foot, right? As well as a cerebropleural ganglion. So another ganglion here, which isn't really like a brain, but it's a concentration in the visceral body mass of the organism, not too far from the mouth. Lastly, it has a visceral ganglion. This is a concentration of nerve materials in the viscera, in the guts, right near the intestines and the exit from the body from the anus. So as I mentioned in the previous slide, this does have light-sensitive structures here on the edges of the mantle, eyes. It's also got these tentacles here for sensory information in terms of touch and chemicals. So it's got the ability to sense light, chemical, mechanical, touch receptors, and probably up and down as well. So you might ask the question, why would these scallops have these eyes, and so many of them, on the edge of the mantle? This is what they look like when you look at them really carefully, when lots of the other bivalves do not, right? And the one thing here is that these scallops are actually much more mobile, and therefore they need to be able to detect shadows to escape. They use their ability to actually open and close their shell quickly to swim, and I'll show you a video of this in class. Here's what it looks like. This particular bivalve, this scallop, is trying to get out of the way of the sea star. It smells the sea star and says, uh-oh, this is dangerous, and off it goes. Tries to move itself as quickly as possible using the power of its adductor muscles. Some clams, including all the clams pictured here in these 4 slides, are enormous. They're huge, they're very sedentary, they are not dangerous like what we see them portrayed to be sometimes. No one will get trapped in them, they can't possibly close that quickly, but they do have incurrent and excurrent siphons, and they're also highly colored. Take a look at the coloration here. We've got this beautiful green, this gorgeous sets of blues, and like some of the other invertebrates, the reason they have these colors has to do, yes, with symbiosis. Right? These are the mollusks that end up forming symbiotic relationships with photosynthetic algae so that they supplement their filter feeding. As we saw in the original slideshow, these organisms have a coelom, which gives them the opportunity for developing the full set of organs that invertebrates at this particular stage of development have. So we have stomachs, we've got digestive glands, we've got a full heart, we've got actually a kidney, right? And we have the muscles that all work to open and close the organism, as well as the foot muscle, which helps it to move, right? So how do they manage excretion? Well, since they have such high volumes of water flow coming in and going out, most of their excretion from the circulatory system is going to be via the diffusion process, particularly with respiratory wastes across the surface of the tissue of the gills. But we see here we have an anus, so any food waste is actually moving through a whole digestive system and then out of the body through the anus. And they also have a kidney here, right, which is going to release the urinary products, the liquid waste products from the organism, and they move out with the excurrent water as well. So the last thing we want to consider is when you are sedentary, when you're stuck in the mud, basically, right? How do you reproduce? What's the best process for reproduction when you are a sedentary creature that lives either on or under the substrate? And if you look here, we can see that on this particular organism, it shows the gonads, right? These are the reproductive tissues, and frequently we have both male and female structures in the same clam. Some of them are separate sexes. If you look here, this is a male clam that's releasing sperm out into the water column, right? So these are broadcast spawners, much like the other organisms that are attached to the substrate. They need to get their seed out into the water. So they're broadcast spawners. The females will literally take the sperm in, and it will make it to the gonads, fertilize the eggs, and then they'll release the eggs in larval form. They'll become part of the plankton, so they have a planktonic larvae which floats around in the water column until it's mature enough to settle down to the bottom and become an adult. And enough about the biology of these organisms, but clearly they are the fodder for many cartoonists, in which we take some liberty with their particular lifestyle here, right, and also making jokes about the nature of clams. Happy as a clam, steamed, right? When they get all steamed up, these are some steamed clams at dinner. There you go. And finally, the next group will be the cephalopods. Far more active, far more dynamic set of creatures than these bivalves, and interesting and very unique in different ways.